Hello everyone, my name is Davide Merulla and I'm really happy to have you here for this third episode of our webinar series. Today we are going to talk about digital staining and best digital staining practice. The beautiful images that you're seeing on your screen have been taken with our 3D Cell Explorer, and the colors that you see have been added thanks to the digital staining. Today, by means of several examples, we are going to show you what is the biological relevance of this digital staining, show you how easy and quick it is to perform this process, what are the physical bases allowing this digital stain to be as selective as the normal type of staining, and at the end of the webinar we will give you some tips and advice to achieve the best digital staining. So as usual we are ready to start, so feel free to go full screen and to use the Q&A panel to interact with us. We will collect your questions and answer all of them at the end of the webinar. The Q&A panel is monitored by my colleague Lisa Pollaro and Luca Clario in the back. So let me start with this first example showing a co-culture of cancer cell and amoebas. Say that, so this left panel here contains the images, the grayscale images collected based on refractive index and collected with our 3D Cell Explorer. You can know more visiting our web page under the webinar section and looking at the first webinar to know how this image has been collected. The central panel here contains a preload digital stain, and this allows me to obtain a lot of information from this image. Say for instance that I'm interested to segment and identify only the amoeba. Very simple. I go here, I activate the stain specific for the amoeba, and automatically all those amoeba are identified in the grayscale image and on our 3D reconstruction. Now, if I'm interested into the cancer cell, I just have to go here, activate the specific stain, and here they appear in both the presentation. So this first example shows you how easy and simple, just by a click, is thanks to digital staining to segment different cell types. And the most important thing is that this segmentation has been done in a completely non-invasive manner, since we didn't have to add any kind of marker thanks to tedious and laborious practice. Now let's get back to our example and try to look at the data a little bit more intensively. I have been able to identify two different cellular types, and later on I want to see how those cellular types interact. Well, very easily, on a data set that I already had, I can apply a third stain, which is specific for the membrane, and this allows me to see how the membrane of the amoeba and the cancer cell and the melanoma interact together. So, thanks to this second example, I show you how it is possible to use the digital stain not only to differentiate cellular types, but also to segment specifically different cell structure, in this case the membrane, that is common to the two different cell types. Okay, so far we've been looking at endpoint measurements. But it's also possible to use our microscope and the digital stain to obtain kinetic information on time lapse. So, in this case, I am interested in monitoring the confluency of my cell line throughout time. I can shoot a time lapse very simply, and I can create a stain. Let me show you again. Can simply apply a stain specific for the mounting media. In this case, I can also scroll through my image and you see that I'm identifying my cells. The stain specific for the mounting media, it applies to time0, as you can see, but it also applies after 30 minutes, 25 minutes, 47 minutes, and so on, so forth. So, very simply, with a few clicks, it was possible, thanks to the digital staining, to identify only the surface covered by the mounting media and plot the surface against time and have a very clear indicator of the confluency of my cell line. So, this third example shows you how the digital stain is also very useful to obtain kinetic information on a 2D image. But as the name suggests, the 3D Cell Explorer is a tomographic microscope. So we can also collect 3D information. What we have been doing in this case was to generate a stain specific not only for the mounting media that you are seeing here, and now for clarity I'm going to silence, but also for the cytosol of the cell in purple and for the membrane of the cell in yellow. What is very simple to achieve as a measurement with the 3D Cell Explorer and by using Steve is to know how much volume these stains is occupying. To do so, I just have to look at my stain of interest, a digital stain of interest, like in this case the one for the cytosol, click on the information button, and this dialog panel will open, and I can see here the volume in microliters. I can just record this number, and as you can see, the same trick applies throughout time. Okay. Now, what I can do very simply is to plot this time the volume against time and see how the volume of the mounting media is decreasing throughout time, the volume of the cytosol is increasing, and the volume of the membrane is increasing as well. I'll let you take the conclusion out of this experiment by yourself. What I wanted to show you, and I hope I was convincing enough, that it's very simple, again in a completely non-invasive manner, to obtain kinetic measurements, but on volumes and not only on surfaces. Okay, so now let's move back to the video that we saw at the beginning of our presentation. As you can see here, thanks to the digital staining, we can identify very well structures into the cell. Like for instance, in this case, we are identifying very well the membrane of the cell. So what we can do, we can follow the process of mitosis of this fibroblast, looking at the thickness or at the change of thickness of the cell throughout the mitotic process. So you see at the beginning and at the end the cell is relatively flat, then it starts to bulge, then it gets to the maximum height, it divides, and then it flattens out again. So thanks to this fifth example, I show you how it is possible to obtain a set of physical marker in terms of phenotypic marker, to monitor the cell cycle of the cell. But there is a lot of information that we can obtain from the same type of experiment, actually from the very same experiment. If we look at this video, what we have been able to do is to follow the condensation process of the chromatin. You see how this cross indicates the value that we are considering, how the color of the chromatin throughout the cell cycle changes, and we were able to identify 5 different regions corresponding to 5 different states of condensation of the chromatin. And again, all of this just in silico, in a very specific way, in a very reproducible way, but just thanks to a few clicks in a completely non-invasive manner without any type of staining. So these 6 examples show you how the refractive index is even useful to monitor some physical changes in the chromatin composition. Okay, you can find all those examples and look at them in a more detailed ways going on our webpage nanolife.ch/application. And I also have to acknowledge all the work done by our bio team, namely Lisa, Luca and Chris, who are the ones who have performed and designed all those experiments and collected all those images. How did they do that? Well, if you are looking at this 3rd episode, probably you've seen already this guy. This is our 3D Cell Explorer, and it's the machine that allows us to collect those images based on refractive index. This is a thing of the present, although it looks like a thing of the future, as I've been telling you already once, and it's available now on our webpage, and you can find it here, nanolife.ch, product 3D Cell Explorer. Now the question is, how it is possible to achieve such type of marking and such type of images? Well, everything starts from the refractive index. The refractive index is the reason why the straw looks broken at the interface between air and water. The refractive index tells you how the light interacts with the specific transparent media, and it's defined as the speed of the light in the vacuum against the speed of the light in the refractive media. The reason why the straw looks broken at the interface is because the refractive index of air and water is very different. In this specific case, the refractive index of water is 1.3 periodic, because it's 300,000 kilometers divided 225,000 kilometers per second. Now, if you imagine that the cell is a collection of transparent organelles having different chemical composition, you can understand how those organelles will interact in a different manner with the light. Hence, they will have a different type, different number, different value of refractive index. It is thus possible to discriminate those organelles based on refractive index and assign a specific color to each organelle thanks to this correlation between color and refractive index value. So, to sum up, the refractive index is a physical marker. It's non-invasive because it's already there. We don't have to add any type of fluorophore or chemical dye. It's highly reproducible because it is the ratio between 2 physical constants. And it's also very quantitative because the 2 constants will be always the same. What do I mean? If we take the example of fluorescence, you know that the value of fluorescence that you measure will change depending on the intensity of the light that you will use to excite it, the time that you will excite it for, and the sensitivity of your instrument. This means that it will vary between experiments and experiments and instrument and instrument. In this case, the refractive index will be constant between all the experiments and constant between all the instruments. So it's a totally reproducible type of marker. Okay, now we are ready to go into the digital image from a specific point of view. And let's start with defining what a digital, an image based on refractive index is. It's nothing else than a matrix where each square contains a value, each pixel contains a value of refractive index. The graphic representation is obtained by assigning a specific color to a specific value of refractive index. In this case, we use a dark gray for low value of refractive index and white for high value of refractive index, and we obtain this type of representation. Now, since it's us who's making this rule, we can decide to assign a completely different color, say green, to a specific value of refractive index, say 2. In this case, we will be highlighting, okay, digital segmentation. We will perform a digital segmentation based on quantitative information on the value 2. Again, since it's us who's making the rule, we can push it even further and silence all the other values. So in this case, we are really segmenting and showing only the information that is related to the value 2. And we have this first type of segmentation. Now, the refractive index is quantitative information, but in an image, there is not only that type of information, there is also local information. What do I mean? I mean that in a background area, the local variation of refractive index will be very low. It will be a very homogeneous area. Instead, a vesicle will be a very small, very different, very high refractive index, will be probably very different from the surrounding. So it will have a gradient which is very high. What is a gradient? The mathematical definition of a gradient is this, but it's nothing else than the comparison of the central pixel with the neighboring one on the on the y-axis, on the x-axis, and on the z-axis. Okay? So we can calculate this value of gradient for each pixel of the refractive index image and create a second matrix. To the second matrix, we can apply a second set of rules. In this case, we are selecting the membrane based on an abrupt change in refractive index, and we are obtaining this type of segmentation. Again, just to not get lost, the refractive index gradient is a derivative information that we obtain from the refractive index that correlates the refractive index to its local environment. But the most interesting part is that we can combine those two rules. So the information, the quantitative information that we obtain by the refractive index and the local information that we obtain from the gradient to increase specificity and move from this loose segmentation to this very high and specific segmentation with very high specificity. Okay, now that we are that we know about the definition of refractive index and index gradient, let's see how to use those information to get the best digital stain. Okay, so first thing first, let's have a mental idea of what this central panel is. This is a bidimensional panel that allows us to discriminate against value of refractive index on this axis and values of index gradient on this other axis. We can divide this panel in 4 areas, and we will have structures like vesicle with high refractive index and high gradient localizing in this area. Structures like nuclei, which still have a high refractive index because they are kind of dense, they interact a lot with light. They are smaller, so they have a more smoother gradient in this area. Structures like membrane in this kind of area, so lower refractive index but high span of index gradient because they will be flanking different regions, so maybe vesicles, maybe nuclei, maybe the background. And the background at the end, so it will be in this area because you will have a low value of refractive index and a low value of gradient. As we said before, it's a very homogeneous area. Okay, so let's start from the background and start from staining the background. So what do? I go down here, push this button here, and I select the color. I like green for the background. And when I start hovering my lens on the image, you see that on the central panel, some pixels are highlighting. When I hold and move around, I'm telling to the software, remember all those pixels. The software is calculating the average of refractive index and index gradient and placing this box exactly at the average. The size of this box corresponds to the standard deviation connected with this measurement. Okay? Now, if I'm not happy with my stain, what I can do, I can manually move it right and left, so to change the range of refractive index that I am selecting, or up and down, so to change the value of gradient that I'm interested on. Now, to better see what I'm doing, so let's restart from scratch so I can throw away my stain like that. Select again the color, create my background and have it there. Another thing that I can do, I can also manually resize my stain. Let's say that this is a stain that I like. I can also silence it for sake of visual appealing. Now, say that I want to identify and stain those little vesicles here. What I can do first, I can increase the overlay. Overlay 100%, I will only see the grayscale image. Second thing, I can zoom in on my detail. To zoom in, I just point and then use the wheel of the mouse to scroll in. Now I select another color, say red, and I paint this vesicle here. What do I, as was expecting, the software is identifying an area here in this region, and if I zoom out and reduce the overlay, I can see that all the vesicles in the cell have been selected, and I can by hand modify a little bit the stain. To have a better understanding of what's going on, it is useful to see the both images, both the segmentation and the refractive in the grayscale image. So I advise you to place yourself around 60–70% over. Okay, I can really follow where my images are. Now, this information is used to create our 3D segmentation. In this case, we can see our vesicles, but they are really pale. So I can act on these other controller, which is the opacity. And I can increase the opacity. The opacity, what does it do, is to define how strongly we are coloring the features that we have selected. The opacity has not to be confused with the edge softness. The edge softness correlates the stain with the pixel, the distribution of the pixel that we have selected. What do I mean? Edge softness 100% will color all the pixels that falls into this area. Edge softness 0% will color only the pixels that are very close to the mean value. So it's up to you define the value of edge softness depending on the feature that you want to identify. Okay, let's go on with our staining. Select another color, say blue, and let's say that I'm interested in our nucleolus. I just hover on top of them and tuck. As expected, my stain will localize in this area. So same type of refractive index, but much lower gradient respect to the vesicles. Now, what I can see is that all those features, if I tilt my 3D plan, lie on the same layer, on the same plan, which is good because we want to avoid artifacts. If I go on, select another color and choose to mark a membrane, like for instance this may be a good representation of a membrane, what I can see immediately is that my digital stain goes underneath the plan of adhesion of the cell, which is not good. It means that we are seeing an artifact. To avoid this, I just have to lift the stain because it means that I am too close to the background. You see that things are getting much better already and we are almost there. Now, the second thing that I can do is to remember that this is a membrane. So it means that it will have, it will flank different type of regions, cytosol, nucleoli, nucleus, vesicles. So I am expecting to observe the membrane throughout a big range of gradient. So what I can do is to select my stain and manually increase the spread of the membrane. Okay, now you see how my stain is getting much better. Now, with a little bit of, this is already a quite good stain where you can see a lot of details, but with a little bit of practice, you can get to a stain like this, okay, where you can really appreciate the different type of compartment of the cell, the complete membrane of the cells, vesicles, nucleoli, vesicles, nucleoli, inner membrane, external membrane, and so on and so forth. How do I know that this is a good stain? Well, first of all I have checked and everything lies on the correct plan. Second, all my digital stains are well separated, so the stain is very selective. I'm really selective for something specifically. And no one of those is touching the background, so I'm sure that I'm not selecting what is not cell. Now, one thing that you should avoid, you see that you can have a lot of information, is to avoid to have monochromatic stain. What does it mean? This that I'm going to apply now is the very same stain, but made using only shades of yellow and green. As you can see here, I am highlighting the same areas, but I'm losing most of the information. Okay, and that's pretty much it. Now that I show you how easy it is to perform a digital stain, how much information you can get from this type of staining, the type of digital staining, and especially how much information you can get without absolutely harming yourself. It's your time to play with Steve. You can do it just by going to our webpage, nanolife.ch/software. You can download our Steve. We are going to have the new release in a few hours, just after this webinar series, and you can start playing with the examples that you have in Steve. Okay, it's all for now. I will be back in 3 minutes time to complete the Q&A session. I'll see you later.